Welcome everyone to yet another Friday afternoon webinar. Today we have Thomas Wolfers here. He holds a PhD from the Donders Institute at the Radboud University in Nijmegen, the Netherlands. Currently he works as a Marie Curie Fellow at NORA, a research center on mental disorders at the University of Oslo in Norway. Thomas's work is focused on utilizing machine learning methods for a better understanding of complex disorders and diseases. It's a pleasure, Thomas, to have you here. Today you will have a talk titled The Average versus the Individual Patient. So please, Thomas, the floor is yours. And I heard also that you will take questions during the talk. If you're able to see them and answer them during the talk, you can do that. If not, you can wait until after the talk. But please, everyone, use the Q&A button or the chat option here. Thank you.
Hello, thank you very much, Klaas, for the kind introduction, and welcome everyone. Today I'll talk about the average versus individual patient, specifically in mental health research. And before I go into some applications of our machine learning work, I will introduce some aspects to the work that are specific to these kind of disorders. And I think many of you may not be familiar with it, but it's important to get a good understanding of the problem that we are trying to address. So first, the history. And there was already in Egypt, in the old Egypt, talks about mental disorders, and in the context of magical forces that possessed people. That in the antique, I mean, of course, this is a rough overview of history. There were so-called psychological or somatic forces or somatic factors at place. They're already considered psychology as an influence. Then the Persians were the first to believe that mental disorders had some organic cause. That was first coined by Al-Razi in the 900 after Christ. And then during this time, we forget that today, but roughly 500 years ago, it was still quite believable that there is something like being bewitched or suffering from witchcraft. And there's an influential book from Weiner in 1550 who addressed that witchcraft does not exist. Until then, it was believed to exist, and after that, it still was. But there was the first counter argument against it. And then eventually people recognized that we should probably treat people that are suffering from mental disorders and not just ignoring them or putting them on the streets. And this happened around the 17th century. But the treatments were unlike the treatments that we have today. And then the early classifications of mental disorders emerged with dementia precox, which is like a precursor of schizophrenia as we know it today, but in a different form. And as the name says already, it is associated with dementia, which clearly schizophrenia is not entirely, at least. And this was around the 19th century. Before, at the same time, we also, so these two happened to coincide together. Mental disorders were considered to affect the brain. And this led to the movement in biological psychiatry. And nowadays, we're working with a classification, with a scheme that's based on symptoms. And this has different versions, and currently we are using DSM-5. This is a rough overview of the history. So when we find a summary of this, we can see that mental disorders are part of human existence since ancient times. Second, that the understanding of those varied with the trends of this particular time, which is interesting observation, I think. Nowadays, we use the DSM and to understand them, and we use the classifications in these books in the so-called case control paradigm, where you have a number of cases that are based on the labels in the DSM, which or on the basis of symptom classifications of the DSM. And in the machine learning terminology, this would be in many studies the labels that we are interested in predicting. However, these labels are unlike labels that you would encounter, for instance, when you train an algorithm to learn the recognition of faces, for instance, because these labels are very unreliable. So, for instance, schizophrenia can be diagnosed in 26 unique combinations of symptoms, and this is one of the labels of the DSM-5 that has the least number of possible combinations. Depression has many more. So in biological psychiatry nowadays, we define a group of patients on the basis of this classification, and we label them as with a specific mental disorder. And we define usually a group of controls that are matched, and then we subtract the two using all kinds of methods. Nowadays, we of course use machine learning because it has a number of positive sides. Before 30 years ago, the majority of case control analyses were based on t-tests or other statistical tests. So what we need for machine learning, of course, is we need a lot of data, and this data should be large in the terms of the sample size. So we need to have a large N. And this is starting to be the case with brain imaging, which is increasing rapidly at the moment. And with this acceleration, we will soon have enough images to do proper machine learning. And we're doing that now. We have large scale data sets at our disposal. And then, as I said, we predict the output, the labels, and these labels are based on the DSM-5 classification in many of the studies. However, I will also argue for why this may not be the best way forward. And then we train algorithms on big computers, so we also have the computers to take care of this vast number of data sets. And bear in mind that the data that you're talking about here is much more high dimensional than what you would usually include into a machine learning algorithm. So handling this data is extremely complex. And then with this computer, this data and these labels, we are hoping to train programs that will tell us something about the underlying mechanism of mental disorders. And this is what people have done in the past. So how did they train these algorithms? They defined, as I said, the group of patients and defined the group of healthy controls. And then they measured a bunch of biological variables. And we did that too. And then you define a decision threshold, decision boundary on the basis of all kinds of algorithms that you can think of, from deep learning to SVM and logistic regression. And then you predict a new case to be healthy or diseased. And you calculate the specificity and sensitivity and accuracy, performance measures, whatever you have or you want to calculate on the basis of how many individuals you predict correctly in an unseen test set. So we have, a while ago now, done a systematic literature review on studies that have conducted this kind of research. So they used all kinds of imaging modalities for the brain, from structural, functional to diffusion, extracted different features on the basis of different definitions of those features, either voxel-based, region-based, or network-based. You have brain networks, which are interconnected voxels that co-vary. And then these features were selected or non-selected and used in a classifier to predict mental disorders or diseases. And then different studies use different validation schemes and different performance measures. However, we in this report only reported accuracy because that was the most prevalent reported measure of accuracy of the algorithm. Having said that, it's probably not the best. So then these were the number of studies at that time that conducted this type of research. And in the end, after the relevance criteria, we ended up with these number of studies included into the study. And what we found was that, surprisingly, first, the field was at that time characterized by relatively small samples. And the accuracy of the predictions varies substantially, from 60% accuracy to 100% accuracy for the smaller samples. And the larger the sample size got, the lower the performance measures appear to be, although these were, of course, a small number of samples. However, in machine learning, this is, of course, very counterintuitive. You would expect the exact opposite pattern in the literature. So this pointed towards a bias in the field, a bias in the literature. And we associated it with, or we interpreted it as a conjunction of a publication bias with mental disorders showing a lot of overlap with healthy individuals. So with people that have a mental disorder show a lot of biological overlap in terms of brain structure, function, and diffusion with healthy individuals. When we conducted our own studies, that's also a while ago now, we increased the sample size and could see the pattern resurface. So actually the prediction that schizophrenia can be predicted on the basis of brain imaging with about an accuracy of 75%. This is of course not clinically relevant, but when I was doing my PhD, I was surprised that this is possible, because schizophrenia is such a heterogeneous construct, and I was surprised that given the measures and given the labels, that it was possible. And then people followed this kind of logic and showed that for various different diseases, disorders, and brain imaging measures, that for some reason the sample size decreased with increasing sample size, accuracy decreased in many cases. And Gail Valoquois associated that with cross-validation failures in these studies. We think also part of the reason for that is a large group overlap, as I said, between cases and controls. So a case control scenario is actually more like you have a group of healthy and a group of controls, and they overlap quite substantially with one another. And this led us to the conclusion that group level biomarkers are not predictive of status, course, outcome, cure of any of the mental disorder. And that's the current status. And therefore, it's probably not the right way forward to just simply look at this group comparisons and link them to biological measures and then say that this is predictive for biological trait of the disease, or this is predictive for the disease or disorder that is complex in terms of biology. Instead, what we are doing, we map the heterogeneity, and therefore we use normative models. Normative models, just to give you a cartoon description first before I will describe the methods in a bit more detail. So you have a growth chart, which is characterized by the height on one axis and the age of an individual on the other axis. And then you can place each individual in reference to this height. So let's say you go to the doctor and you have a child, and the child is a little bit smaller than what would be expected on the basis of the norm of that for that population. Then the doctor might say, oh, we should probably look at disease or something, or maybe nutritional problems or something like that, because the child is unexpectedly too small for his or her age. The other scenario might occur as well, where the child is larger than one would expect that. We do that for the brain. So instead of using age, but we used age usually, we predict the size of the brain in different shapes and forms and places and locations using machine learning. And then we not just do that once, but we do that 1 million times across the whole brain. And then we create a space for which in which we can place each individual. So each individual then lives in a multi-dimensional space where we make predictions of what the person's brain shape is in a particular location across the whole population. And then you can look at each individual on how it deviates and responds in reference to these different brain locations. And here I would like to talk about, first of all, this is Andre Marcontes, my PhD supervisor when I was in Nijmegen. And here he showed that you can model these norms in different ways, of course. So first you can use a linear model, which doesn't reflect the population well. You can use a nonlinear model, which then you can model the mean appropriately, but that wouldn't be enough if you want to give information about how much an individual deviates from that mean proportionately. Then you could model the variance terms in different fashions. And ideally, you want to have a model that's nonlinear and also model the uncertainty appropriately. And in most of our studies so far, we used a Gaussian process regression approach, but we also used deep learning and other things. So how do you then use these models? So you have a statistical model where you have a growth chart and you want to detect whether one individual deviates, and you use all kinds of covariate measures to predict the brain shape, as I said. And then you get nonlinear or linear decreases or increases of this particular brain location in response to this. And you do that for all kinds of brain measures, so cortical thickness, age, and age as covariates input, and you can do that for all kinds of features on the brain side. So we have brain structure, DTI, function, etc. And you use a kind of validation approach to estimate the norm. You estimate the norm using cross-validation in a large population, and you validate it out of sample in this way. And then once you've trained the model on your healthy control cohort, on the population cohort, you apply it to the target cohort and then look at how individuals in that target cohort deviate from that norm. And you not just do that for one normative model, but for multiple. And then here I show you one application that we did. So we used the normative model to describe the brain aging process in various different, in a healthy individual. So what you see here is a forward model of such a model, plotted the mean of the forward model, plotted against for males and females, and the difference between males and females. So what you see, generally speaking, is that for males and females, you see a similar decay of gray matter. In this case, gray matter is a measurement that we took for quantifying the brain structure in a specific location. And we see that the gray matter decays or decreases in frontal regions and in the cerebellum and in the insula. And what this says is basically that males tend to have a slightly larger brain than females, and so forth. And we did not just do that in the discovery sample, but we did that across 2 replication samples. So we recovered the similar kind of behavior of the normative model across 2 replication cohorts. And from the age 20 to 70, in some regions, we see a decrease of 0.2 in brain volume. And these are maps for those working with brain imaging. These are based on BBM maps, voxel-based morphometry maps. So then once you have established such a norm, so such a normative model, and for the different brain locations, you can look at how and where each individual patient deviates from that norm. So instead of looking at group differences, you look all of a sudden at how an individual deviates. So you can think of it, this is, for instance, patient 1, and patient 1 shows thresholded significant deviations in these brain regions. While patient 2 with the same diagnosis, so all these patients have a diagnosis of schizophrenia, show a different pattern of deviation from that brain norm, from the brain normative process. And we can do that for patient 2, 3, 4, 5, 6, and so forth for all the patients in the group. And they will all show some similarities in terms of how they deviate from the norm, but also a lot of differences. And here we show a classical, we just took it back to the classical case where we show a difference between between a healthy group and a control group, which is done in most studies or in all disciplines, in most disciplines that address clinical clinical relevant complex disorders. We find a pattern that's in coherence or that supports the general gist of the literature. So we find multiple comparison corrected differences between healthy individuals and schizophrenia patients in these regions of the brain, and this is replicated across 2 studies. Largely replicated. We see some slight differences, but most of the differences between the group differences are replicated. And this is true for bipolar and schizophrenia as well, but to a lesser degree. Our replication there is a little bit less clear. But of course, the classical statistical tests with an increasing sample size, you are more likely to find larger differences, and this sample is smaller than this sample, than this sample. And this gives you an indication for what is usually reported in the literature on brain structure described as a difference between healthy individuals and schizophrenia patients. So when we now look at the individual patient overlap maps, so I showed you earlier how individually the patients overlap, we observe this pattern. So what we did here, we looked at within the schizophrenia group, what are the brain regions where more than 2% of the patients with schizophrenia deviate extremely from the healthy norm. And we see that in some brain regions, we see more than 2% of the patients with schizophrenia deviating extremely. And these brain regions tend to be similar across studies. Yeah? However, this is 2%, so it's not like the majority of the patients deviate extremely in those brain regions. So by looking at the group average effect, we hereby show that it's very hard to say anything about how an individual deviates from the norm in response to that, or as a consequence of showing a group average effect. And for bipolar disorder, this pattern is even a little bit more distinct. And these are extreme negative deviations, so all the deviations are extremely negative from the norm. Mariam, she did work on autism, where she looked at the age range from age 6 to 30. She described the normative model now not on brain volume, but on brain structure, on brain thickness. And she showed the norms for these different regions and could show that typical developing controls show less overlap than autism patients, extreme positive overlap, but only in 6% of the patients at the max, or individuals in these groups, indicating again that there's extreme heterogeneity in the ASD cohort, or indicating that there's extreme heterogeneity in the ASD cohorts as well as for the extreme negative deviations. Then Johnny, he used an approach normative modeling approach to detect white matter lesions in the brain. So here you can, of babies. So what you see here, you see infants from week 24 to 40, and he trained a model, normative model on these infant brains. And then you see here the mean absolute error of that model, so where they can predict well how the brain looks in the normative population and where it's difficult. And then he looked at which voxels would indicate the white matter lesion. A white matter lesion can happen, for instance, when a baby is born and they have blood flow issues or certain damages to the brain, then you would see white matter lesions. So it's extremely relevant to detect those. And then you can see here how well he could detect the white matter lesions using this approach. For instance, here you have the raw T1 image of an individual baby. And then you have the lesion masks. So this is the ground truth. People went through this and checked where there are white matter lesions, trained experts with a lot of experience knowing how the brain should look like. And they detect white matter lesions, so white spots in the brain where there shouldn't be white spots. And then he calculated the Z-scores with the GPR model, or just on the basis of that scoring. And he used a cluster threshold, and then he could detect almost perfectly all white matter lesions in this particular group of infants. And Rally, she used a similar approach to use on DTI on diffusion measures. And diffusion is how water molecules travel through the brain, and then from that diffusion matrix you can derive different measures of, for instance, FA, which indicates how directional diffusion is, or mean diffusivity, which is like the average diffusivity of the water molecules in the brain. And they generate pictures that look like this, and the FA is clearly linked to fiber tracks, so the white matter bundles in the brain, because white matter bundles are they are protected by a lipid layer, and the lipid layer is not penetrable by water, and therefore the diffusion is more likely to be directional. This is what you in principle see on these images. And then she used a normative model to model this effect in infants. And then you can look here at individual infants and in the degree of deviation from the norm. And if here, this is an infant that deviated in 12% of the voxels, of diffusion voxels, and this is one that deviated only in 0.6. And then you see where it deviated and whether there's a pattern and so forth. And now, another very important aspect of this work is, of course, in order to derive the norms, you really have to work with big data. And Mosi is working on this. He developed models to deal with big data sets, because then you have to deal with a lot of problems that you don't deal with in smaller data sets, which may be acquired in one sample or one scanner. So in this case, there are this number of scanners. I wouldn't count it now, but many scanners in the sample, and there's a huge sample size. And then you, of course, have to model the differences in scanner effects. So one scanner that acquires data in Oslo is different from a scanner that acquires data in Bergen. And you have to account in your model for this difference. And he modeled that in different ways and compared these methods. So here we can look at the results of this comparison. So for instance, I would take out combat. Combat is a method that is generally linked to the gold standard at the moment in the field. So where people use, it originally comes from genetics. So in genetics, you have batch effects for ships with which you sequence the genome. And in order to get rid of this batch effects, geneticists developed the combat method. And then recently, like 2 years ago, 1 year ago, this method was adapted to brain imaging data. And here, Mosi shows that the HBR approach in the context of normative modeling. So if you use the HBR approach, it's better than, for instance, the combat method. And the combat method is basically you regress out side effects. And in HBR, you model the side effects in the normative modeling framework. And then he could show that that patients with schizophrenia, bipolar, and ADHD, so forth, could be predicted using his model. So this wraps up the content of the talk with respect of the current work. And now I'd like to talk shortly about the future. So in future, when, for instance, you have a family member, maybe your name is Vincent, and you have a monozygotic twin, and you live in a family full of artists. And one day Vincent gets sick, he develops psychotic symptoms, and then he goes, or he or she goes, Vincent in this case, he goes to the hospital, and then the hospital has access to a large anonymized data set, population data set acquired across various biological measures. And then the hospital's access to machine learning facility that derives all kinds of norms and normative measures, and then you can think of different scenarios. Or let's say you have a million norms, and then a family which is closely genetically linked and related, and then you look at individual deviations from that norm in reference to the population. So there could be a scenario when the whole family lives within the norm. So then the combination of these biological measures is unlikely to be disease causative for Vincent, because otherwise he's within the population norm, and schizophrenic or psychotic symptoms are not very prevalent in the population. If he would be the only person that lives outside of the norm, then these combination of biological markers are likely to be potentially causative for the disease, and therefore would need to be explored in more detail. So that's what potentially could be done in future with this. So to conclude, the dominant trends at the time determined the understanding of mental disorders at the time. When relatively recently, or so in the context of the, let's say, the last 30 years, which is not very recently in current time, but in the larger context, machine learning methods have been used to predict to predict mental disorders on the basis of different brain features. And this is important because we can integrate a lot of information using these methods. And as the disorders are complex, it is definitely a way forward. However, it appears to be not the only thing that we have to do, because case control comparisons have shown us that while we show robust group effects, we often observe little individual level overlap, which is a problem, because if we show a group effect, we know little about the individual patient while we know something about the group. Therefore, we need to refine our understanding by mapping the heterogeneity within a group. And for this, I made a case for normative modeling and how that could be done, and showed a couple of examples where we did it. With this, I'd like to acknowledge a lot of people that have trained me or that I've worked with. And in particular, I'd like to point out Andrew McCond, who has been my PhD supervisor, Christian Beckmann, and Lars here in Norway, with whom I'm working at the moment, and everybody else. And I might acknowledge, I might have missed people, but there are many people, and it's great to work with all of them. And hereby, I would like to thank everyone for listening in, and hope that you liked and have a lot of questions. I'm happy to answer them.
Thank you so much, Thomas, for a really interesting talk. So everyone, please use the Q&A button or the chat if you have any questions. I can start, Thomas, if that's okay. To me, it looks like it's really hard to use these biomarkers, at least as a diagnostic tool. I mean, there are a very small fraction of people that have prominent deviation from the normal population when it comes to schizophrenia, for example. So my question is, for these people that have kind of this, it was like 5 or 6% that was quite different. Could they have any individual treatment, or what's the difference? What can you kind of do with your with your results here? Could you provide a medication, or could you, for those cases, provide a diagnosis, or how could you use the results?
Yeah. This is a very important point, and this is a matter of current research. So we have figured out over the last decade, let's say decade and a half, that these group effects are not reliable, and we have shown that now quite clearly, or are reliable but are not giving us information about the individual patients, and therefore we need to dissect the heterogeneity within that population. Of course, once you have a dissected phenotype, you'll be able to give more targeted treatments, because you more accurately define the disease. Or to speak in terms of machine learning, you would know, your label would become more precise, and therefore you would likely also to detect better treatment targets.
Yeah, I will continue with the question from Jon Becker. Could normative models of age be biased by society? For example, young people use a lot of technology now, but older people use less technology at the same age. Does this show up in these models?
Yes, I mean, you can build these normative models on all kinds of data points. So if you would be interested in, for instance, building a normative model for the usage of technology, you would build a population norm of the usage of technology, and this population norm should be reflective of the whole population. So you would use a random sample from the population that you're interested in, then you train these models for age, and then you would definitely find a difference in terms of extreme usage in the younger population versus less or even extreme negative usage or extreme less usage in the older population. I'm sure you would detect this, and yes, this is broadly applicable to these kind of questions as well.
I also know there are some projects about actually the use of cellular phones as more or less not a diagnostic tool, but at least a tool to predict certain mental disorders. Are you aware of these projects, or is this not in your group?
Yeah, of course, I'm aware of this. So this is certainly super interesting, I think. So it's called sampling. So if you sample like in the daily environment of an individual, and in terms of our work, that would be a novel kind of features that we could include into the model. So certainly that's possible. If you have a measurement of a mobile from a mobile phone that's interesting for mental health or any other disease, then you could use a normative model to detect whether there are deviations from that over time, for instance, and so forth. So definitely interesting and definitely important. And it would be the feature step in this modeling process. So the way you derive the features.
We have some more questions from Just Janssen. How sure are you that deviation, that could be brain structure or function, are clinically relevant? That is not due to some other factor, for example, image quality.
Yeah, that's an important point of consideration. So in our studies, we associated the deviations with all kinds of clinical measures like PANSS scores, etc., as well as image quality measures. And yes, some of the deviations will be due to image quality, but as has been shown in the study, for instance, with the babies, where we knew the ground truth, the white matter lesions, where the ground truths that were to be detected, the methods can detect things that you're interested in, but it's definitely an important priority of research to detect deviations from normative process that are disease relevant versus those that are not.
Exactly. And from Julian Mayer, hi, very impressive talk. Do you expect that to diagnose mental disorders, you have all the right data at the disposal, structural, diffusion, functional, or are there blind spots, obvious data that you expect would contribute to better fit?
Yes, I think that's a very important question. We have to basically, in my opinion, explore various different feature sets. So from the ones that I talked about here were brain features, but there may be other features that are relevant that may indeed be linked to mobile phone usage or other stuff. Behavioral features. It might also be that it's genetic features, but genetics is it has to be modeled differently than brain imaging. Because genetics doesn't evolve. It's something that's there. It's building blocks, and then everything that happens from there is going to determine the structure and function of the cells and ultimately the individual. But I agree, all these factors need to be considered carefully in the modeling process. And we certainly have to explore the feature space. There are interesting studies, I haven't talked about that yet, that used EG, which seems to be quite interesting if you use that in a different way. And then there are studies that looked at speech patterns, which showed very interesting results for schizophrenia, for instance.
And another question here from just Janssen. Would it be better to include patients plus controls in the large normative model?
It is best to include a population sample. And patients are, of course, part of that population to a certain degree. So ideally, and that's where we work towards is to build normative models that are representative for a population. That's the work that I've shown from MOSI, where we build large-scale normative models that are then more representative for the general population, and that would be ideal. So patients would then be part, if they're part of the population, that would be part of that sample, but they shouldn't be part of that sample to a large degree, because schizophrenia occurs in the population about 1%. So exactly. It shouldn't go in, yeah.
Exactly. Yes, and a question from Katarina Jørgensen. Do you think this would this be of any potential use in predicting which medicines are useful for which patients?
Yeah, that's the ultimate goal, but I think honestly we have to work a lot more to reach that goal. I think that's the ultimate goal to get, is to become treatment relevant, that we can find subtypes or subgroups or sub-individuals within the schizophrenia population that respond better to a certain treatment versus others. Yeah, that's the target.
Yeah. So how is that? I don't know schizophrenia at all, almost, but it's now treated as kind of one disease, or is it also now medicated and treated differently between groups, or how is that actually?
Yeah, it is the case, but usually what happens is you give one medication, you figure out whether it works, and then you give the next, and so forth and so forth. Okay, so it's kind of errorless, like an iterative process, and yeah, and for some individuals you don't really find a treatment that helps, and for many you find treatments that work after testing them, but then they have a lot of side effects and so forth. So it is important to find better targeted treatments for, and I'm not talking just about schizophrenia, I'm talking about most of the complex disorders and diseases here, where we don't have very targeted treatments available. And then it's always a combination of, do we have the right treatment, and do we give that, if we have the right treatment, do we give it to the right individual? And this work is then targeted towards finding the right individuals. But the right treatment is the question. That's another question, whether that exists at the moment. So that's another, but that's not part of that research at the moment.
But you have done also studies very correlated the treatments that actually works, and try to find which kind of patient groups that works for, or is that?
Yeah, we're working on that, but the problem here is that you need to have really carefully selected samples that would allow you to do this kind of work. So that's ongoing work. Yeah.
Exactly. Yes. Yeah. Many thanks from Julian Mayer. I don't think we have any more questions now. If anyone has a question, you could still ask. We'll see if it comes in now. If not, I think we'll just call it a day and say thank you so much again, Thomas, for a very interesting talk. And we will also have a webinar next Friday. Then it will be, we alternate now. So next Friday it will be NORA Startup, which is kind of our innovation network, but still AI and still relevant. So please tune in 2 o'clock next Friday as well. And thank you, Thomas. It was a great talk.
Thank you very much. I'm very grateful for the opportunity. Thank you.
Thank you, and thank you to all the attendees. See you next week.